Hello, everybody. Today I'm going to present evolutionary stability of receptor binding domain of SARS-CoV-2 spike protein. As introduction, since early 2020, more than 10,000 single mutations have been detected in the SARS-CoV-2 genome worldwide. Intelligent studies have shown that a specific mutation in the SARS-CoV-2 spike protein can change the structural stability and recognition affinity by the ACE2 cell receptor. In particular, receptor binding domain of S protein is crucial to enhance the infectivity of SARS-CoV-2. Furthermore, N-terminal domain of S protein contributes to changing AC2 RBD conformation, and its variability serves to evade the host human response. Our objective is to determine the evolutionary stability of the receptor binding domain of SARS-CoV-2 protein. As method, we use a database of NCBI and preprocessing 39,457 complete sequences of SARS-CoV-2 spike protein. Then we alignment the sequences in and with our algorithm, we retrieve the fields the first time each mutation was detected. Then with Discovery Studios, we perform each mutation. After that, with Ghost Pro Server, we make the molecular docking, and finally with a segmental model, we use ligand receptor binding energy corresponding to each mutation that were plotted. As a result of discussion, we can see amino acid variability SARS-CoV-2 and SARS-CoV-2 reverse sequences. We can see that RBD has greater variability and could be the region that is modified to infect other species, namely that it is crucial in this evolutionary adaptation with the human host. Therefore, should be some combination of mutation that produces stable conformation between RBD and AC2. Then we can see mutation frequency plots of variance shows where amino acid change occur. We can see 4 mutations and 1 region were seen with the highest rate of point mutation: A344, B367, L452, A500 2020, and A475, E484. Then we can see regions of maximum variability in RBD sequences and variability motifs represent the interior structure of RBD. 70% of mutation occurred during the first quarter of 2020. The reach in RBM contains 54 of all mutations that could be associated with an adaptation to enhance recognition of the N-terminal helix in AC2. Then we can see ligand receptor binding energies corresponding to each mutation date. Our results suggest an adaptive stability trend that is associated with a higher affinity energy in RBD AC2 recognition along that timeline. This reflects an evolutionary adaptation that is independent of the geographical region in which mutation arouse. The alpha, beta, gamma, and delta variants have at least 50 mutations. 2 E484Q and L452R in the RBD. Finally, it is necessary to continue with genomic surveillance of mutation that allows making inference about the infectivity and lethality of new variants that arise in the future. As a conclusion, the genetic diversity of SARS-CoV-2 is likely to increase as it continues to circulate in the human population, so phenotype max should become increasingly valuable for viral surveillance as mutation accumulate. The overall stability of RBD contributes to the binding affinity for AC2. In general, the mutation effects on RBD binding and expression are correlated to the high pressure of positive selection observed. This is leading to variants with higher affinity for AC2 for higher infectivity conjugate with the antibody-mediated immune response induction. Thank you for your time.